Welcome students. Today we are going to talk about a plant tissue culture paper. This program is for BSc third year students, subject botany, semester 5th. Paper code is BOD 101. Title of today's unit is Unit 1, Introduction to Plant Tissue Culture. Module name, Sterilization Practices in the Laboratory. Module number 05. I am Nehal Asgaonkar, Assistant Professor, Government College of Arts, Science and Commerce, Cape Goa. The outline of today's module, I'll be talking about the concept of sterilization. What are the different methods of sterilization used to sterilize the plant tissue culture laboratory? There are three different types. First, we'll be talking about using disinfectants. Fumigation techniques is the second one, and third one is using sterile cabinets with HEPA filters. Learning outcomes. This module will explain the concept of sterilization, and it will also explain the various methods which are used for sterilization in the laboratory. Concept of sterilization. Before learning what is sterilization and why do we need sterilization, let's learn what is contamination. Contamination is the presence of a constituent, impurity, or some other undesirable element that spoils, corrupts, infects, or makes something unfit or makes something inferior, a material, physical body, natural environment, or a workplace, etc. There are three main sources of contamination. First, through culture vessels and culture medium. Sterilization of these culture vessels and culture medium is done using autoclave. Second, you get contamination through explants. Sterilization of these explants is carried out using various chemicals, and sterilization of this explant is also known as surface sterilization. Third is seen during inoculation. So this inoculation procedure should be carried out into a closed area with HEPA filters, and this is carried out to the transfer room in a lamina airflow unit. Cares to be taken during inoculation procedure. Ensure that the laboratory is dust free to avoid contamination. Wash your hands before handling any cultures or reagents. Use proper clothing that are aprons and headgears. Check for contamination in the media bottle before using them for the experiment, and do not leave any culture bottle open after inoculation. So now, what is sterilization? This is a method by which all the microbes, spores and acellular entities, which includes virus, viroids, pyrons, are destroyed or removed from an object or an habitat. So why there is a need for sterilization? Sterilization eliminates microorganisms and thus avoid contamination by bacteria and fungi. To maintain an aseptic environment, all culture vessels, medias and instruments which are used in handling tissues as well as the equipment and the explant should be surface sterilized. How can microorganisms be killed? By denaturation of proteins, that is by breaking of many weak linkages and bonds, which includes the hydrogen bonds. Then second is by interruption in their DNA synthesis or DNA repair. And third one is by disruption of their cell membranes. So there are various methods of sterilization. Two general methods are physical method and chemical method. Under physical method, you have the heat sterilization, which includes the dry heat method and the moist heat method. Dry heat method is done in a hot air oven, whereas the moist heat method is seen in the autoclave. Then we have the filter sterilization. We use different cellulose, different pore size filters to sterilize different chemical reagents. And third is physical method, which is done by radiation. Under chemical method, we use different chemicals, for example, ethyl alcohol and sodium hypochlorite. Methods of sterilization of the plant tissue culture laboratory. In this, the whole area which is there for the plant tissue culture lab is sterilized. This is done using disinfectants, fumigation techniques and using sterile cabinets with HEPA filters. First technique is using disinfectants. Disinfectants are nothing but chemical agents which are used to inactivate or destroy microorganisms on surfaces. Some examples are alcohol, formaldehyde, potassium hydroxide, phenolics, sodium hypochlorite, ethanol, ammonium compound. Commonly used are ethyl alcohol and sodium hypochlorite. Fumigation techniques. This is the type of gaseous fumigation wherein vapors are taken. Method of gaseous sterilization in which It is used for killing of microorganisms and prevention of microbial growth in air, surface of wall or floor. This method completely fills an area with gases fumigants to suffocate or poison the microbes and pests within. Procedure of fumigation. There are two methods seen. Method 1 is using electric evaporator with formaldehyde. As we all know, formalin is available as 40% formaldehyde in water. Effective at 20 degrees Celsius and relative humidity as 65%. This is the instrument in which vapors are formed. Method 2 is by using formaldehyde with potassium permanganate. This is carried out using a evaporating dish. 35 ml of formalin is added to 10 grams of potassium permanganate. You will see purple fumes are generated. What are the precautions to be taken before and after fumigation? You have to ensure that you clean all the windows, doors, floor walls with soap and water. You have to ensure that there are no openings to the doors and windows. You have to seal all the doors and windows. Switch off all electrical devices. Use proper clothing, that is apron, and proper head gears and even glasses. Paste a warning notice on the main door for restricted entry. Advantages of fumigation. Fumigants are toxic to all the microbes. Therefore, it is possible to control all life stages of microorganisms. It is the quickest way to sterilize the given area. It reduces any residual problems. Third type is seen using sterile cabinets with HEPA filters. These sterile cabinets are also known as lamina airflow units. These are present in the transfer room of the plant tissue culture area, wherein all aseptic transfers or inoculation take place. High efficiency particulate air filters in the LAF cabinet. Lamina airflow can maintain a working area which is devoid of any type of contamination. HEPA filters are designed to control the number of particles entering a clean area by filtration. How do these filters work? HEPA filters to use two different types of mechanism. First, there is one in which there are outer filters which work like sieve to stop the larger particles of dirt, dust and hair. Inside this, there is a mat of very dense glass fibers designed to trap very small particles. To summarize this module, contamination can be controlled using a technique of sterilization. Sterilization is a method of killing of microorganisms in a material or an object or an entire area. These are some references. Thank you.